That's good.
Okay, very good. Thanks for the invitation to talk. Haven't seen Peter Nair in a long time. It's wonderful to hear his presentation and progress. What I'd like to tell you about today is a very different type of vaccine against Streptococcus pneumonia. It's a vaccine we call MVX-01, which is a protein fusion antigen. And it's a fusion protein between a very potent toxin that's been toxoided called pneumolysin, fused to choline binding protein. And this is the whole concept of a very different type of vaccine, which is serotype independent. So this is first and foremost a bit about who Matrovx is. We're privately held by a group called Morningside Ventures, located in the Boston area, in Boston specifically. We've taken one program, which was a typhoid fever vaccine, very successfully through phase 1 trials. And we are seeking a partnership on that. And we've now sort of defocused our typhoid vaccine, Typhax, and we're going after some more robust markets, in particular, like I said, the pneumococcal vaccine candidate MVX-01, which I'll tell you about in the next 20 minutes or so. And then we have a very interesting vaccine candidate, C. diff, against Clostridium difficile. So this slide here is entitled, and I think appropriately so, the evolution of pneumococcal vaccines. So much like the problem that Peter identified about different strains of rhinovirus, well, there were different strains and serotypes of strep pneumonia as well. So we're now really at 100 and counting. And these strains really encapsulate themselves with polysaccharide, and that's the basis of the serotype nomenclature. It's also the basis of how they avoid the immune system by resisting opsonophagocytosis. So it really is kind of an interesting way of categorizing the strains and serotypes. So as I mentioned, there's 100 and counting. If you go back to the beginning, so to speak, Merck still has a vibrant market for Pneumovax. That's 23 individual capsules that are unconjugated. So it works very well in, let's say, 5-year-old to 50-year-old persons, highly fairly immunogenic, but certainly nicely protective. But again, because it's unconjugated, really has no value in children under the age of 2, and in particular with immunosenescence, it's of questionable utility or efficacy in the elderly. So what first evolved was Prevnar 7. So that was 7 of the 100 now capsules, but the ones that were at high incidence and high virulence, and that was introduced in the year 2000, 90+% efficacy in the United States, and it's certainly a wonderful benchmark for a new vaccine that protected the most needy, right? The very, very young. Synflorix of GSK came on with a 10-valent. It's only licensed, I should say it's not licensed in the US, but it's licensed throughout Europe. And really it's been what we call the capsular arms race since then, from a 7 to a 10-valent to Prevnar 13, which is the current most sophisticated vaccine that has 13 valencies. Pneumosil is a Serum Institute vaccine that has a different subset of capsules that targets more the developed countries. And now, again, we're really close to getting licensure of a 20-valent by Pfizer and a 15-valent by Merck. It's kind of interesting, a local company to us, Affinivax, was one of the first companies to start thinking about adding protein components to polysaccharide vaccines to basically get a greater breadth of protection. So these are some of the highlights I've already talked about. Like I said, the 100 serotypes. It's interesting that the serotypes cover 3/4 of the clinically relevant strains in the developed world, but far less so in less developed countries. And there is this phenomena of serotype replacement that's quite real. That is, if you vaccinate against a certain serotype, because the nasopharyngeal area is such a wonderful place to live, it will be populated by sort of the next up serotype that begins to get a foothold and take on greater incidence and even virulence for that matter. So I'll spend a minute here, but no doubt in terms of value, or the pneumococcal vaccines are the most sophisticated. They're also the most valuable. So you can see the global sales here on the left bar graph, and I'll focus on that because that's the composite of the US and rest of the world. And you see just for Prevnar alone, they were around 6 billion. For Pneumovax, still roughly $1 billion, and Synflorix about 600 and so million. So really, this is a $7+ billion a year vaccine market with expectations to grow, certainly even over the upcoming years. So now begins to talk about our approach towards making a pneumococcal vaccine. So unequivocally, the 13-valent Prevnar 13 is the most cumbersome, arduous vaccine to make. It's 13 individual conjugations. You purify capsule, you purify a carrier protein molecule called chromosome-97, different conjugations, and you sort of cocktail together. So we're going after a serotype independent approach. And essentially the main molecule, the key player here, is a toxoid of Pneumolysin. And we have two mutations, one in the ligand site and one in the pore site that prevents that locks the antigen into a pre-pore configuration. That's amino acid changes at residues 293 and 460. And then we fuse to that in a single polypeptide two different antigens that are derived from CBPA, which is choline binding protein A. One is a highly conserved sequence that mediates binding to human polymeric immunoglobulin receptor, and the other, another highly conserved sequence that mediates binding to the laminin specific integrin receptor. And what these antigens and epitopes do is the pneumolysin, as I mentioned, that is clearly the hallmark virulence factor of strep pneumonia. It forms pores and cells, and I'll show you a beautiful photograph of that in a minute. It's not actively secreted, but it sort of leaves lyse cells of strep pneumonia. It's kind of a mystery as to how the toxin actually leaves the cell, but it's highly conserved and highly lethal. And then again, on the flip side over here, CBPA, these two epitopes, as I mentioned already, are ligands and help the organism to actually go on in virulence and pathogenesis from the nasopharyngeal area all the way to the meninges. So again, more about pneumolysin itself. It's in a class of toxins called CDCs, or cholesterol-dependent cytolysins. It's 30 to 50 monomers. You can see a picture of the pore that it makes. So these are the individual monomers making a 25 nanometer pore. It targets neutrophils, very important in its pathogenesis. Once invasive pneumonia develops. As I mentioned, highly conserved, produced by nearly all clinical isolates, very important virulence factor. And I'll show you antibodies to pneumolysin are highly protective in a murine model of infection. The CBPA, it's a protein adherence factor, often a ligand that again helps to continue invasion from the nasopharyngeal area and beyond. There's many different clades. It's highly disparate and mutagenic in some areas. And we've cloned two of the conserved sequences, what we call the peptides I've already mentioned to you, CBPA A1 and A2. That again, I've already mentioned to you, have these different roles in tissue invasion and dissemination. Okay, so here's how we see our vaccine work. So if you go back to the conjugate vaccines in particular, their area of efficacy is really right up front. So they really preclude and prevent nasopharyngeal colonization. So in fact, what it looks like, that's what the new SARS vaccines are doing too. In a recent study, they will actually reduce transmission. So because you leave this area uncolonized, something else can come in and colonize, typically another strain or serotype of strep pneumonia. So our anti-CBPA antibodies will help against acute otitis media and upper airway colonization. And then we're relying on anti-pneumolysin antibodies and as well anti-CBPA antibodies to again prevent or preclude dissemination from the lung into the heart and into the meninges. So pneumolysin and CBPA, again, just to kind of bring the two together, major virulence factor, highly cytotoxic. It is fortunately immunogenic. And CBPA, again, a ligand, so we're going after secreted toxin and a ligand. And we've cloned the most important or conserved areas of the antigen. What we like about this approach is that it's very inexpensive. It's a single polypeptide that we make recombinantly E. coli, roughly 55,000 Daltons, and it should lead to a substantially decreased lower cost of goods. Okay, so here's some of the evidence that supports our thinking for this vaccine candidate. There's several studies, and for example, the first one in Papua New Guinea, where it was shown that anti-PLY maternal antibodies delay early carriage in high-risk New Guinean infants. So here's where there's no vaccines. And when you look at protected offspring or progeny of these moms through placental transfer, and what you in maternal antibodies, you get a sort of an association of much less disease carriage. And there's also other studies published by Holm and Zhang Mendy that actually also support PLY and CBPA. So these are the two antigens that antibodies against in newborns are associated with lesser disease carriage and lesser disease. Another very important fact as we think of one of the opportunities for our vaccine candidate is a maternal immunization strategy. And it's very interesting that a study was done with a 9-valent PCV in women in their last trimester. And when they looked at tried to immunize the offspring, the children, with Prevnar, there was a decreased antibody response. So this was blocking antibody against the vaccine. So we see a wonderful opportunity in high-risk areas for our vaccine to immunize mothers, and again, leave the child protected early. And then when they begin their immunization regimen against a capsule-based vaccine, that could actually happen without any interference. Okay. Another piece of data that I think supports our argument quite well is GSK had a differently toxoided pneumolysin. It was actually chemically detoxified in a histidine triad protein, and they immunized macaques in a study, and with their ASO2 adjuvant, and of 34 macaques, they got antibodies against each type of antigen. And with a challenge with a highly virulent 19F serotype strain, 94% of the vaccinated monkeys survived, and 62 with the adjuvant alone. Again, this is good supportive data for our projects. And then clinically, it's nice to know that Sanofi has tested one of their versions of a pneumolysoid with or without or with combination of other antigens. And they get very high immunogenicity at a relatively low dose of 25 micrograms. And there was a recent study by GSK with their PLY antigen and PHT, the histidine triad protein, and they get an incremental protection against acute otitis media, but very interestingly never studied invasive pneumococcal disease, where we think our antigen would have really its greatest value. Okay, so just a little bit of nomenclature, because I'm going to show you preclinical data from some of our different precursor constructs and constructs that we have. So there's a single mutant that reduces cytolytic activity of pneumolysin by 5,000-fold. When we couple it with the other attenuating mutation, we cannot detect any activity whatsoever, less than 125,000, so undetectable. There's an antigen that one of our colleagues and collaborators uses, YLN, which is a single mutant with the CBPA peptides. And MVX01 is the preclinical candidate that Matrovx is moving towards the clinic. And that only is different from YLN by 1 amino acid. So it's important to recognize that. Okay, so these are some of the early studies that we had done at Matrovx, asking the question, is this idea of a serotype independent protein vaccine, is it worthwhile? So we asked Steve Pelton at Boston University to give us a lot of disparate strains that weren't lab passage, clinical isolates, highly virulent from different areas. So we really asked a very tough question early on. And these are some of our better results that we got. But as you can see, we can get, in some cases, complete protection with a highly lethal challenge with these different serotypes. Some of these are contained in Prevnar. Some of them are not contained in Prevnar and emerging strains. And like I said, in some cases, it's really quite binary, as you can see here, where you get complete kill of unimmunized animals and very high protection. And again, there's this, the same is true as 6A. All our animals, 10 of 10 live versus 0 of 10 survived after 50 hours. So we knew that this strain could protect against relevant clinical isolates that we, again, got from the Pelton Library at Boston University. We then asked the question, how did our vaccine work against serotypes that were comprised in Prevnar? So we went through a like dosing regimen, a three biweekly dosing regimen, and immunized with either Prevnar, a naive group, or our pneumolysoid fusion, the double mutant. And again, what we saw here, and these are very aggressive strains. This is an 18C strain. We had 100% viability of our mice in 10 of 10 mice. And as you can see, even the animals that were immunized with Prevnar, because our inoculum is so high, it's 100 million colony forming units, all the animals died. And here's even a more lethal strain, a serotype 4. You can see we got partial protection, but again, very few of the Prevnar immunized animals survived. So in this particular case, we could overwhelm a Prevnar immune response, and these serotypes are included in Prevnar, and show that our vaccine was actually superior in protection. In this next piece of data, we took even a more virulent strain of 6B and asked the question, how did that behave either alone with our double mutant antigen versus Prevnar, or the two together? And we really see a very nice synergy slide here. What we see is, first of all, our double mutant, which is in red, gave a very high level of protection, or longest time to death, particularly compared to Prevnar, which is in blue. This is the control. But in this kind of fluorescent colored bar graph here, forgive me, this line graph, what you can see is we've got synergy showing that some protection, if you will, upstream in the nasopharyngeal area, plus against invasive disease, we actually got roughly 40% of our animals to survive. So our vaccine works in complement or independent of a Prevnar vaccine. And this is just a quick review of the data that we have. We tested 28 different strains representing 20 different serotypes, and collectively we got about 85% protection. Again, these are serotypes that were contained within Prevnar or beyond Prevnar. This is some factoid, so to speak, about some strains that weren't virulent or were hypervirulent. And then we got superior protection against the two challenge strains we tested, 4 and 18C, against Prevnar comprised strains. And again, I just showed you the data that show that the antigens can actually work synergistically as well. Okay, so this is a piece of data that was generated from our collaborator, Elaine Tuominen at St. Jude. And YLN, as I mentioned in that nomenclature slide, that's only a 1 amino acid difference from MVX01. It's just one of those point mutation that attenuates. So it's essentially the same. And she did a very extensive series of studies to look at the difference of the benefit, the added benefit of these peptides, in this case, CBPA2. Remember, one of those peptides is a ligand that's only recognizes human polyimmunoglobulin G receptor. So we cannot score its benefit here. We can only look at one of the epitopes, CBPA2. And if you look at the otitis media model, you can see alum as a control. The POI mutant really didn't do much, confer much level of protection at all. But as you can see, the percent otitis media dropped substantially when the CBPA peptides were added to the L460. This bar graph and these photographs here on the right really kind of show a comparable story. Here we're looking at infection within the lung in this panel on the right, as well as the development of meningitis. And as you can see, again, the alum control, we got a high degree of bacteria in the cerebrospinal fluid. L460T was indeed helpful there. And then when you add these CBPA peptides, you get a further reduction with that's statistically significant. And then on the right panels here, you can see this is a saline control. So these animals were not challenged. And the YLN vaccine regimen really looks quite comparable to that. Very different from the alum control of the animals that were actually challenged. And this shows cellular infiltrate that's actually evident in the alveoli of the lungs. And again, the L460D did actually quite well, far better than the positive control, and but not nearly as well as the POI mutant with the CBPA peptides. This is a study that we did last year, prepping to move our vaccine candidate into clinical trials. This is the results of our tox study, where we went to as low as 10 microgram doses up to 90. We did include one group that did not have L-hydrogel, but as you can see, there's a really nice dose response. These are just ELISA titers here against GMTs, against POI or CBPA. And you can indeed see that as the dose regimen increases, the dose response profile is really very nice. Not too much augmentation here between no L-hydrogel and some, but there's perhaps a bit of a difference that you can see reflected after 2 doses. And again, if you look at CBPA conversion, pretty comparable across the board. And we didn't see too much of an impact, at least on the ELISA titers that are shown. So in toxicology, we had no systemic or injection site reactivity at the highest dose regimens or dose levels and regimens. And I can tell you that from clinical data from Sanofi, 25 micrograms is highly immunogenic. So we're certainly well beyond that target dose. And then in seroconversion that I've shown here. So these are looking at some functional antibodies now. And as you can see, this anti-hemolytic titer, this ability of antibodies in titrated fashion to neutralize wild-type toxin and actually form that pore in red blood cells. And again, you can see a very nice dose response. Not too much of a difference here between the unadjuvanted and adjuvanted controls. And then here we're looking at percent adhesion. Again, this is only reflective of that CBPA2 peptide 2. And our best level of interference or precluding adhesion, if you will, was with the adjuvanted group at the 30 microgram dose. And it's interesting that the unadjuvanted group really had a high level of attachment. So perhaps we're going to see a greatest impact of alum adjuvanting on the CBPA peptides that are part of our vaccine. So getting close to a sum here, so a wrap up. So MVX-01 clinical development. As I mentioned earlier, MVX-01 is a recombinant fusion polypeptide expressed in E. coli. We are making our material GMP in the next couple months, and we aim to file our IND in the second half of this year. This is a general look at our trial design. Again, it's classic safety and immunogenicity at different dose levels. We're going to start out at 10 micrograms with the placebo with group sizes of 20 altogether, 30 micrograms, 60 micrograms, and then 90. And then we're going to do an open label and a blinded study, double-blinded study in elderly subjects. So once we see the best tolerated in a nicely immunogenic dose level, that dose will be tested in the elderly who we think will benefit most from our vaccine approach. In summary, I showed you data that non-human primate challenge study supports the pneumococcal protein antigen vaccine idea. Our own data that we've collected showed murine immunogenicity and protection data against the really a bevy of different Prevnar 13 serotypes and emerging serotypes, where in many cases, as shown here, we got actually superior protection than Prevnar 13. And we showed a nice synergy here with a 6B strain challenge. MVX-01, which is our molecule of interest, highly protective against lethal pneumococcal challenge. And we showed that in mice. And as I just mentioned, our clinical plan is really focused on the elderly who do not do well, not well protected by the current polysaccharide-based vaccines. And with that said, I'll take any questions if you have any.
I request the panelists, to the attendees, to please raise their hand, and we can proceed with the Q&A session. So we have like 5 minutes, and then we can go for a 10 minutes break.
Okay.
So we already have a question from Farshad. So Mr. Farshad, if you can come online, unmute yourself. And if you would like to directly chat with Mr. Kevin, you can do that. Or else, Mr. Kevin, you can see the query in the chat box.
Oh, there's a question there? Okay.
Yeah.
Hi, great talk. Just wondering if the dose of alhydrogel is the same in rodent and non-human primate and human, or do you adjust by the weight basis?
So yeah, so the non-human primate study, that was not ours. So it's a good question. I do not recall what GSK used. No, sorry. The non-human primate study, they used ASO2. So that's a combination of like a saponin and an MPL derivative. So that was from a GSK study as supportive data to our program. So the only studies that we've done with alum or without are in mice and in rabbits.
Have you ever tried to combine it with CPG?
That's a study that we're actually planning to do. We have an adjuvant in-house from a collaborator, and we are going to try a CPG adjuvant, yes. But we have not as of yet.
Okay, thank you very much.
Thank you, Mr. Farshad. So we have next query from Maria Gurza Pizza.
Yeah. Hi. Yes, can you hear me?
Yes.
Yes,
Very well.
okay. So very nice presentation. Congratulations. Very, very nice. So I have two questions. One is about the protocol of immunization you are going to use in humans. So how many doses and the distance between the different doses? And the other question is also, are you going to do any non-inferiority with the classical conjugate vaccine in your clinical trials?
So those are very good. The first one's much easier to answer than the second one. So the first question is, our dose regimen in rabbits were 3 doses, 0, 14, and 29. But you always give an extra dose in rats. So our plan for a clinical trial is day 0 and day 29. So it's a 2-dose regimen in our human studies at those dose levels you saw, 10, 30, 60, and 90 microgram levels. And we think we're really going to be again, if the Sanofi data are a good indicator, and I think they will be, we're predicting perhaps peak titers at 30 and perhaps 60 micrograms. But it's nice to know that we have that window to go higher, particularly in the elderly.
Yeah.
Non-inferiority. So we are now looking, first of all, we can't look at surrogates, right? Because with a surrogate would have to show anti-capsular antibodies that match the titers of Prevnar, and obviously we have a whole different type of antigen, so that goes out the window. So we're looking at places now that have unusual serotypes that are not included in these, the valencies. In fact, I said it's Prevnar 13, but a 15 and a 20 valent vaccine
Yes,
around the horizon. So where there are places in the world that do not have, those sorts of serotypes that are in play. And it's quite feasible to do a study that's a non-inferiority study if we have to, again, because of the prevalence of the different serotypes.
Thank you. Thanks.